Today, we'll be discussing a developmental network of the thorny dragon, where we'll be able to predict whether the thorny dragon skin cells will produce spikes or will not. Let's get into it. In developmental pathways, arrows indicate activation. Hence, the protein to the right of an arrow is assumed to not be present unless its activator to the left of the arrow is. Horizontal T-like structures indicate inhibition of a pathway. Hence, the protein to the right of a T-like structure is assumed to be present and be blocked by the protein on the left if the left protein is present. If the protein on the left is not present, it would not be able to block the protein on the right. Thereby, it could perform its biochemical action. Now let's look at a more complex developmental network to apply our knowledge. Here we have S activates P blocks I blocks K activates E, and E activates spikes. Depending on which proteins are present or not present, spikes could be produced on the skin cells of the thorny dragon or not produced. Now let's look at some interesting cases where certain proteins may not be present in this network. In the wild type of this developmental network, S activates P, therefore P can block I. Since I would not be present, I cannot block K, therefore K can activate E and E can activate spikes. In the absence of P cannot block I, therefore I can block K and K cannot activate E, and E cannot activate spikes. Hence, spikes would not be produced. In the absence of I could not block K, therefore K could activate E, and E could activate spikes on the thorny dragon. Now let's look at what would happen in the absence of I and E. Since I is not present, I would not block K. Even though K would be present, we already have said that E was not present. Since E is not present in the mutant copy, spikes would not be produced. In the absence of S and K, S could not activate P, therefore P could not inhibit I. However, since we already said that K was not present, E could not be activated, and thereby no spikes could be produced. In some instances, skin cells of the thorny dragon may not have spikes, while some skin cells may have lots of spikes. Let's look at a process known as DNA methylation to see how this process could result in the outcome of the thorny dragon having spikes on its dorsal side, but not its ventral side. DNA methylation is basically just the addition of a methyl group to specific regions of DNA. This epigenetic regulation helps to suppress the production of a certain gene. Since all cells of the thorny dragon have the same genome, this epigenetic regulation could explain the differential expression of the spikes between the dorsal and ventral portions of the dragon's body. Let's assume that methylation occurred to the DNA that makes up gene E only in skin cells that make up the ventral portion of the thorny dragon's body. If this were the case, gene E would not be transcribed, and therefore protein E would not be found in the developmental network for skin cells that make up the ventral portion of the thorny dragon's body. However, in the dorsal skin cells, gene E is not methylated, therefore it can be transcribed and translated into protein E, and therefore spikes will be produced.